Jen, why don't we get started because we have a lot of folks and we can keep admitting people as they arrive. Welcome, everybody. I'm Mark Strama. I'm the director of the Annette Strauss Institute for Civic Life at the University of Texas at Austin, and I am the chairman of the board of the Happy Lungs Project. Thank you all for joining us today. The mission of the Happy Lungs Project, as you probably know, is to save the lives of RET-positive lung cancer patients by advancing research and treatment options. We do that by funding and facilitating cancer research and educating and empowering patients to participate in clinical trials and lung cancer registries. We are thrilled to be able to share some of the updates that you're going to hear today from researchers who have received funding from Happy Lungs Project. I want to thank all the doctors and researchers who are joining us to share this important information with us. Thanks to Lily for making this webinar possible, and thanks to everyone who has supported Happy Lungs Project in this effort. Now I'm going to turn it over to Irene, our Director of Research at the Happy Lungs Project, to introduce our panelists.
Thanks, Mark. Hello, everyone. I'm pleased to introduce our panelists today. So the first one is Dr. John Heymach, Professor and Chair of the Thoracic Head and Neck Medical Oncology Department at MD Anderson Cancer Center. Second one is Dr. Alexander Drilon, from Thoracic Medical Oncologist and Chief of the Early Drug Development Service at Memorial Sloan Kettering Cancer Center. The third one is Dr. Justin Gainor, Director of the Center of Thoracic Cancers and Director of Targeted Immunotherapies at Massachusetts General Hospital. And the last of the speakers is Dr. Alexander Reuben, Associate Professor in the Department of Thoracic Head and Neck Medical Oncology at MD Anderson. We are very grateful you are all here to present all these updates with us. So just a few housekeeping things before we started. One is that we are going to record this presentation so that we can share it afterwards. If you have any question, please put it in the Zoom chat, and we will ask a few questions at the end of the meeting. But for any questions that don't get answered today, we will ask the investigators afterwards, and we will get back to you as soon as possible. So with that, we can get started. It's my pleasure to introduce Dr. John Heymach. So if you are ready, Dr. Heymach, please feel free to share your slides.
Okay. Great. Thank you, Renee. Do you have the right view here?
Yes.
Okay, great. Well, so I'd like to start off with thanking Happy Lungs, for the support and, just emphasizing, what I've seen through the years is that progress for subgroups of lung cancer really ends up getting driven much more by individual groups like Happy Lungs than sitting around waiting for the field to do it. I've sat on the major National Cancer Institute panels that decide grants, and with the grant funding as tight as it is right now, only about 10% of submitted grants are getting funded. It means there's typically a motion just to focus on the major cancer types, and it gives short shrift to the genomic subgroups like RET. So it's really groups like Happy Lungs that are the ones that are responsible for getting momentum going. And I think RET is a great example of one that Happy Lungs has really had a big impact on the progress we're making. So I'm going to talk about structure-based assessment here of the RET receptor and genomic mechanisms of RET sensitivity and resistance. And again, happy to take whatever questions people would like, and forgive me if I end up lapsing into jargon a bit, but okay. So I think everybody here is aware that that for RET, we've got two FDA-approved drugs. We've got selpercatinib and pralsetinib. Those are both very good drugs. They're ones that we use routinely, and I think have really had a major impact on the field. But just to highlight a couple of points. First of all, we're treating RET fusions here like it's one thing. But really, when we say RET fusions, what we're talking about is a group of genomic alterations that we think results in the cancer. But they're a very heterogeneous group. There's a lot of different RET fusions. Just to remind everybody what a fusion is, fusions occur when a chromosome breaks, and one gene, the RET gene, is broken in half, and another gene, which can be a whole bunch of other genes, is broken in half, and the two joined together. So genes that are not supposed to be together are put together, and that causes RET to get turned on. But because there's a lot of different places that break can happen, and a lot of different partners that it can fuse with, we've got a whole variety of different RET fusions here. So when we look at this group, and this is showing all the tumor responses, so if the bar goes down, that means the tumor shrunk. Well, you see almost all the tumors are going down. The drug is causing shrinkage in almost all of them, but the degree of reduction varies from 100%, all the way to, just about zero. So tumors are not responding similarly. So what my group is focused on is can we understand why there's a lot of heterogeneity in response? Why do some people respond well and some people don't respond well? And then when the drugs stop working, can we predict that instead of waiting for individual people for them to stop working and then trying to deal with it then? So that's a big picture here. And, just to give this example, this is from a big data set of RET fusions here, almost 500 RET fusions. And if you look at when that gene breaks, the most common thing it emerges with is KIF5B. So for anybody here out there, that you've got a KIF5B RET fusion, you see that's by far the most common, but we see all these other partners here as well. And some of these partners also partner with other oncogenes. For example, TRIM24 can break and not only join with RET, but it can also join with BRAF. I have some patients who have TRIM24 BRAF fusions that are causing, resistance. So from studying both sides of the partnership here in a fusion, we can learn things that may be relevant for other tumor types as well. Now, with this said, one source of heterogeneity are these different partners that the chromosome can join up with, but even where the chromosome breaks is different as well. So, the first way to study this is a little bit of cataloging here. And you can think of this as, if you've got a new species of, some type of animal, and you put a bunch of people out there to try to identify every variation of that animal and start studying them. Well, this is a little bit of the type of cataloging that we're working on doing, which is taking every type of RET fusion that can occur and making a genomic model of it, which then lets us study different drugs, different ways that the tumor can become resistant, and so forth. So we've been doing this with Happy Lung support, and we do this in a model called BAF3 cells. Now, BAF3s are a very commonly used model. It's a type model where it won't grow on its own unless you put a cancer-causing gene into it. And so here you can see, we've made, now more than 20 different fusion partners. And for KIF5B, the most common one, we've even sort of reproduced each of the different breakpoints, a 1512, 1612, 2312, and so forth. What that's referring to is what's the exon in KIF gene and what's the exon in the RET gene where the break occurs, and then they join. The exons are the little units of a gene, so that tells you exactly where in the gene that the break occurred. But you can see this sort of full catalog of different types of fusions. And once we've reproduced these genomically in our models, it means we can test any drug against any of these different fusion partners. Then it becomes a very simple experiment to do. Now, you might say, well, why not just collect them from different people? Well, we certainly want to do that as well. And collecting different fusions from different people is very, very important. But any given person has not only KIF5B, but they've got a lot of other factors that could impact the response. And we want to understand those, but part of what we want to do is isolate the effect of this RET fusion itself and say, all right, if we just look at the RET fusion, can we figure out how these different fusions respond? And then do we need to make new drugs that would work better for some fusions as compared to the others? Now, we have some other lung cancer models here as well that have been taken from patients, and we have a growing list there as well that we're working on. So this is all sort of building the tool chest so that we or other investigators can study which drugs work for different RET fusion patients. And this is, to our knowledge, I think the biggest collection of different fusions. Now, the models, we believe, are pretty good representatives of what happened in patients. So, for example, we have a patient-derived model from our lab, the MDAL tumors, and it's a KIF5B RET fusion. It's a 1512 in this case. And then we have sort of the one that we genomically engineered on the left. And here we've just shown the effects of, 7 different RET inhibitors. And, if you sort of look at the drugs the way to read these curves is if you see this is measuring how many tumor cells are left at higher concentrations of the drug. So the less drug it takes to kill the cancer cells, the further the line is to the left. And if you look here, you see the gray and the green box lines are the ones furthest to the left. That's pralsetinib, selpercatinib, and ponatinib. And then from the patient model, you see the exact same order. So in other words, we can reproduce with our genomically engineered models exactly what's seen from a given patient's tumor. But we also then can make any genomic model and have the same background cells. So it really lets us compare apples to apples across the board here. So one of the key things we've learned is different RET fusions have different drugs that are effective. So in other words, so instead of saying every RET fusion patient should get selpercatinib or everyone should get pralsetinib, what we're learning is different fusion partners have different responses to different drugs here. And if I give the example here of the CCDC6 fusion, that's one represented with the orange line. If you see selpercatinib, selpercatinib does not inhibit that one very well, and you see the line is higher. But if we look at pralsetinib inhibits it very well on the right. So this is, and cabozantinib not as well. So this is an example where if somebody walks in the door with the CCDC6 fusion, the recommendation would be, well, maybe we should use pralsetinib instead of selpercatinib for that. Now, that's an observation that had already been made in the clinic, from treating patients, but it sort of confirms that our models are able to detect what's being seen with patients. So this is really building the catalog of models that we need to then be studying all these different drugs. And the way you can think of this is, you'd ultimately like to be able to put together a big lookup table where you say, for this particular fusion, here's the drugs that work, here's the drugs that don't work. Now, this is a big heat map, and what this means here, every single box here on this means that we tested in a number of experiments that particular fusion with that particular drug. So this is thousands of hours of experimentation all sort of represented here. But if something is bright green, that means that the drug works very well against that fusion. If it's red, it means it doesn't work against that fusion. And you can see here that for different drugs, there are cases where there's a lot of green, but there's cases where there's a lot of red here. And if you take that CCDC RET fusion that I mentioned before, you can see the little black box for selpercatinib. Again, the greener it is, the more potent the drug is against that fusion. So it's not very potent against the CCDC6. But this lets us test new drugs as they're coming along and seeing which ones they can inhibit well or not well. Okay, so that's sort of a starting point. And now that this resource is available, this lets us take any new drug that comes along. But what I'm not showing here is we're also working then on using molecular modeling to say, all right, do we have ways to improve the effectiveness of different drugs against specific fusions? So instead of looking for one drug for all RET fusions, can we pick one subgroup of the RET fusions and try to inhibit that subgroup? Now, a second part of the reason we want to build this library is the following. We want to also be able to understand when a tumor becomes resistant to RET inhibitors, how it becomes resistant. Now, one way we can do this is wait for people to become resistant. But first of all, that takes a long time. And secondly, if you're waiting until somebody's tumor becomes resistant, you've lost ground already. And so what we want to do is understand if we can sort of accelerate that process. And, you know, we broadly group RET inhibitor resistance into two big categories: RET-dependent resistance, which means the RET fusion itself changed. So we just might need to find a different inhibitor that could block it. So those are mutations in RET itself. But the second is what we call RET-independent resistance, where the RET fusion may stay exactly the same, but something else gets turned on. And if we can develop ways to predict what else is going to get turned on, we may be able to block it before it ever emerges. You know, so in other words, if we can develop the tools to say, well, here are 3 or 4 things that may happen, and here's how we can look out for it early, then we can say, well, can we target those cells before they actually become resistant as well? And so let me give the example. Now, before I go on, I should have paid attention. How long do I have? I want to make sure I don't push everybody back here. Renee, what's the total time for me?
The total time is 15 minutes.
With 15 minutes total?
Yes.
Okay, so I should be done already then, pretty much, right?
I think you have 5 minutes or so.
Okay, all right, good, thanks. Okay, so I want to give this example here for people that are familiar with evolution and you know, what Darwin first described on the Galapagos Island with the finches. And what he observed is there were, you know, this was a place where it was very isolated. Presumably there were 1 or 2 finches, 1 or 2 pairs that originally were taken there by a ship or flew there. But then over time, they evolved a tremendous variety of different beaks and other adaptations that would help them pull out seeds from different places or you know, get at different food sources. So essentially, you started with 1 finch and put evolutionary pressures, and then you got evolution, which was driven by mutations to lead to different adaptations. But there are, of course, many, many different ways that finches could evolve, right? So, you know, here are all different finch evolutions that all started from a common progenitor there. Well, now, if you think of RET like this, and you think of these cancer cells trying to survive, and they're facing the pressure of a RET inhibitor that's trying to kill them, well, RET, you know, can mutate in different ways that could enable it to survive. But there's not infinite ways. There's many ways, but there's not infinite ways. And we know if we, you know, wait for patients and just collect and occasionally get that data, we'll detect some of those ways. But what we wonder is there a way we could sort of scan all the possible ways and understand them before they occur, even if they never end up occurring, instead of just waiting for what actually occurs in people? So the way we've done this, and this is in collaboration with Ralph Kittler, our collaborator at UT Southwestern, is an approach called lentimutate. And, you know, normally evolution proceeds because DNA makes mistakes when it gets copied. But when it gets copied, it makes mistakes at a very low rate, you know, a couple times every billion copies that are made. But if you're waiting for evolution to happen with that very low mutation rate, you got to wait a really long time. So instead, we use a virus that's been engineered to copy the DNA, but it's been engineered to be really bad at copying the DNA. So instead of making a mistake once every couple billion nucleotides it copies, it might make a mistake every 1,000 or every couple hundred times. So we call this an error-prone an error-prone lentimutate virus. And what we do with this is then take the RET gene and we let millions of copies of it get made. And with those millions of copies, we can be confident that every single possible mutation that can occur occurs, but instead of taking tens of thousands of years to wait for it to happen naturally, we're inducing this artificially in a couple days. So now we can look at millions of different mutations all at once, and we can say out of all those millions of mutations, which ones cause resistance? Now, it isn't to say that all millions are going to occur in patients, but out of the ones that occur in patients, there'll be a subset of these millions of different mutations. And so with this screen now, we've been taking the common RET fusions and then putting it through lentimutate, and then saying which mutations survive. So we put in 1 million mutations, we treat them with selpercatinib or pralsetinib, it kills almost all of them, and we say which ones are left behind, and then we look at those resistance mutations. Now we can learn a lot from this. One thing that we see, these little bars that are sticking up, we call these lollipop plots, these show us the frequency of different mutations that led the cancer cell to survive after we do lentimutate. And so just from this one experiment, we're able to identify all the resistance mutations that have been identified in selpercatinib-resistant patients, but it took us a couple days instead of 5 years of study, which is what it took the field to identify them. And so we can do this for selpercatinib and we can do this for pralsetinib. But what we can do is actually much more detailed, because we can do this for every different fusion partner. Now, the data set we have in the world is just from all the patients that are treated in the world that have been reported publicly, and how they became resistant to the drug. But in one case, we may have somebody with a KIF5B 1512 fusion or a CCDC6 RET fusion, and we might have a couple of different mutations reported for each of those. We group those all together. But here we can go fusion by fusion and come up with the full library of resistant mutations. Then what we can do is say, all right, if you've got this resistant mutation, we can go back to all the RET inhibitors that have ever been tested and say, all right, maybe here, we can pick one of these mutations like the G810S that causes pralsetinib resistance. And then we can say, all right, we know that you're resistant to pralsetinib, but which drugs work against that mutation? And the usefulness of this is over time, as this data set develops, well, it should provide patients with an opportunity that says, all right, selpercatinib is not working or pralsetinib is not working. This mutation occurs. Do we have any other drug that we already know that works for that mutation? And so here we've identified a bunch of different mutations, and some of them cause mutations to both selpercatinib and pralsetinib. Some of them cause resistance to just selpercatinib. Some of them cause resistance to just pralsetinib. And when we see these mutations, we can engineer them back into the BAF3 models I was showing before, and then test them. So, now we've developed what we think is sort of the largest data set of understanding all the mutations that can cause resistance to RET, at least for the common fusions, like the 1512 KIF5B RET, and then sort of a catalog of what drugs can work. Now, our goal here is not just to have that catalog, although that catalog is very useful, but then to say, okay, what are the unmet needs? What are the cases where we have no drug that works? And then we can go back to the molecular modeling and say, all right, how do we modify one of these existing drugs so that it could work for, a G810S mutation or, a Y806N mutation there as well. So this is all sort of laying the foundation for that next generation. And, giving an example here, Here we're showing the baseline KIF5B 1512 fusion. That's in black. You see selpercatinib inhibits it very well. That black line is to the left. But when we put in a G810C or a G810R or S, all of those cause a resistance there. But now if we go to the heat map on the right, and we look at that, there are some drugs like the ON12330 and the zetletinib that work pretty well. You see the bars are green there. So we would say, all right, if we had that, these are the drugs that don't work, these are the ones that do. And from molecular modeling, we can start figuring out how could you design a drug that works even better from that as well. So I think the key point here is we can show different mutations and selpercatinib versus pralsetinib, which mutations cause resistance to one of those two. In some cases, you could switch from one drug to the other. In some cases, you have to go to entirely new drugs as well. And, one of the things we've seen here is that next generation inhibitors, and we've listed some of them here, can target these different resistance mutations well. So if you look here at our KIF5B RET mutations that cause resistance, you can see here some of these newer drugs that actually look very effective against these mutations. So this is sort of the catalog that'll let us really compare apples to apples across all these different drugs as well. All right, so I've used up my time, so I haven't really gotten to talk about the RET-independent mechanisms, but let me just say we're looking at the different pathways that are getting turned on, and then coming up with combinations to target those other pathways also. And I want to just highlight one in particular. It's a target called CD70. I think Irene is very familiar with this because she was working us when this was originally identified. And we see when cells become resistant to EGFR inhibitors, we found that CD70 gets turned up, and we could develop things to target CD70 to wipe them out, especially on these cells that are left behind, what we call the drug-tolerant persisters here as well. Well, what we see in RET is these same things get turned up at resistance or in the drug-tolerant persisters. And so we're now investigating approaches for targeting CD70 and this other protein called AXL, so that when you take a drug like selpercatinib or pralsetinib, you may kill 98% of the cells, but we want to figure out how to finish off that last 2%. And that's what we're trying to work on with these targeting approaches, is how do we target those last 2%, what we call these DTPCs, with different approaches like CD70 and AXL, using either CARs or antibody drug conjugates. And I'm happy to get into all the details there, but I want to respect the time here. So just a few key points. We now can see very clearly that different RET fusions, either that have different partners or different breakpoints, have different sensitivities to RET inhibitors. So you can't just pick one size fits all that will cover all RET inhibitors. And then the cells that are left behind that don't die after a RET inhibitor have different proteins on their surface, and we can target those proteins to try to wipe out those residual DTPCs, which we really consider to be the enemy, because those are the cells that left behind that end up growing up and becoming the resistant disease that eventually emerges. So we don't want to wait for it to emerge. We want to come up with strategies to target it before it comes out. And that's the sort of direction we're eager to move in. So once again, I'd just like to thank Happy Lungs for their support. And sorry if I ran a couple minutes over, happy to take questions.
Thanks so much, Dr. Heymach. Now we are going to hear from Dr. Alexander Drilon. Alexander Drilon, please, if you're ready, feel free to share your slides. You have 15 minutes. Thank you so much.
Thank you, Irene. And likewise, thank you very much to Happy Lungs and everyone that's been involved with the group for the support for this project. And thankfully, Dr. Heymach has already given you the beautiful setup for this research that we're doing on targeted therapy resistance. And in addition, I'm going to talk about how active or not active immune therapy is in patients whose cancers have RET fusions. So as you all know, I'm Alex Drilon from Memorial Sloan Kettering Cancer Center. I'm one of the co-leads of this project. Dr. Jessica Lin from Mass General Hospital has really played a pivotal role here. And so I am presenting on behalf of the both of us and the many other doctors and other team members who have helped with this. So we're going to skip this slide and go to the two major aims of this project. And as you'll see on the left, the first aim is to take that knowledge that we heard about regarding resistance to existing drugs and see if this really carries over to what we see in the clinic with our patients and with what we're observing on biopsies of cancers that have developed progression or growth on selpercatinib or pralsetinib, or even liquid biopsies. So looking at any cancer DNA that's floating around in the blood. And the second aim is to look at how effective immune therapy is for RET fusion positive lung cancers and RET altered tumors in general. So I'll start with the first piece. Again, Dr. Heymach had set up the introduction here, so I'm going to go much more quickly. We are calling this project the Registry. Many of you have heard this from before. And the whole point here is to try to aggregate the experience from patients around the world. In the middle, you'll see that we were specifically looking for any cancer that was dependent on RET. Lung cancers are dependent on RET fusions, as are some types of thyroid cancer, like papillary thyroid cancer. And in contrast, medullary thyroid cancer, a different type of thyroid cancer, relies on RET mutations primarily. We were looking for patients who were treated with one RET inhibitor at least. This could have been a multi-kinase inhibitor or some of the older drugs you heard about from Dr. Heymach, or a selective inhibitor like pralsetinib or selpercatinib. And of course, we were looking for patients who had had either a liquid biopsy or a tumor biopsy. So that was for the first aim of the study. For the second aim, we were looking at patients who had received immune therapy either by itself or with chemotherapy. And you'll see on the left the scope of the project, where we were grateful for the participation of many patients around the world at the different centers that you see here. Certainly, we weren't able to cover every country, but the fact that this was a global effort was really phenomenal. Moving to the next slide here, we had set up a central database, and I won't go through all of the details, but that database is called REDCap, and this was a project that was approved by the institutional review board committees at each site. And this provided not just the approval for it, but a mechanism for all of the sites to send their data via the REDCap database centrally for us to look at. Okay, and while you saw the map of the different centers involved, we also wanted to show you the actual people who are involved with getting us this data. You'll see here representation from many centers in the United States, in Asia, in Europe, and Australia. So really wonderful participation from all of these places. And this is an update of how many cases that we've accrued so far. So we have almost 100 patients for whom we have data. And from those patients, recognizing that there were patients who may have had a biopsy before they got their targeted therapy and after the targeted therapy. Here you're seeing that there were a total of 118 distinct TKI resistance biopsies. 14 of these were rebiopsies, like I mentioned, of either tissue or plasma at the same time point. And of the 118 samples that we had, most of them, or almost 100 of them, as you see here, 99 were resistant to selpercatinib, and 19 were resistant to pralsetinib. And you'll see the breakdown there of tumor versus liquid biopsies. I think this in a way also reflects some of the preferences or practice patterns in terms of what drugs are prescribed and received by patients. And it does appear that many providers do tend to pick selpercatinib, even though pralsetinib is also a great drug for this setting with really good activity, as we've seen. So here we have the characteristics of the 99 patients. And I'll just summarize this and say that this is reflective of what we know about patients with RET-dependent cancers. So you see here that many patients were diagnosed at a younger age than we would typically diagnose someone with a lung cancer that's related to smoking. In that same vein, you'll see that many patients actually never smoked or maybe smoked a little in the past. The predominant cancer type was lung cancer, specifically lung adenocarcinoma, but we did have thyroid cases, both the medullary thyroid cancer type, or MTC, and other thyroid cancers like papillary thyroid cancer. You see the most common fusion type or mutation type listed here, and the drugs that were used. So to cut to the chase, here you'll see the top line results of the resistance that we're observing in these samples. And what you'll note is that in the blue slice of the pie, in 12% of cases, these cancers appeared to continue to be dependent on RET. However, they became resistant by acquiring a mutation that interfered with the binding of either selpercatinib or pralsetinib to the RET protein, and thus interfering with its ability to shut down cancer growth and signaling. We'll go through that in a little bit more detail. But in the yellow slice of the pie, this was the last part of Dr. Heymach's talk. A little more than half of cases, we observed that the cancer, while probably remaining dependent on RET, sort of phoned a friend and activated another protein in the cell that was not RET. And we'll show you what these proteins are, but these are other proteins that can be strong drivers of cancer growth. And in the gray slice of the pie, in a little over a third of cases, we still have unknown mechanisms of resistance, which this is a group for which there's really an opportunity to develop novel therapies. And you heard from Dr. Heymach that they're looking into AXL and CD70. And so the hope is that we close the gap here and find new treatments. These are the different mutations that mediate, the technical term is on-target resistance, but it just tells us that these cancers, while these new mutations may have developed, are likely still reliant on RET. And the practical point here is that these are cases for which we can probably look to a next generation RET inhibitor, meaning patients can start with pralsetinib or selpercatinib and potentially hop onto another drug that was designed to inhibit these mutations. And as many of you know, there are several drugs that have been tested. Fortunately, the ellipsis drug, the exploration of that is still ongoing and active. And we have seen cases of cancers that have acquired these resistance mutations that have then responded to the EP0031 drug, which is reminiscent of some other targets in lung cancer, like EGFR, ALK, and ROS1, where you might start with one pill and in the right context, be able to switch to another pill and extend the amount of time that we're able to control the cancer. This is very technical on the left, but just showing you diagrams that illustrate that point I mentioned earlier about how these mutations functionally set up blockages in the RET protein that make it very difficult for a drug like selpercatinib or pralsetinib to bind. And thankfully, these newer drugs like the ellipsis drug and hopefully other drugs that come down the pike are designed to circumvent this and still bind the RET protein and shut it down despite the blockages that are put up by these mutations. I'm going to skip this slide. Here we looked at whether or not some of these major resistance patterns were associated with how long patients were on their first RET inhibitor. And the punchline here is that the duration of therapy did not seem to be different in patients that had a RET mutation versus those that did not. So maybe in more simple terms, it doesn't mean that if you're on selpercatinib for shorter versus much longer, that there may be a dramatic difference in the likelihood of acquiring one of these resistance mutations and thus being eligible, say, for one of the next-gen RET inhibitors like the Ellipsis drug. So that was one of our observations. We are taking this further and looking not just at a single biopsy of a single site or a single time point with a liquid biopsy, but Dr. Lin from Mass General Hospital is looking at cases of patients who have very generously said they would agree to an autopsy so that we would be able to look not just at a single site to study resistance, but look at many different sites of cancer to ask the question, is it the same type of resistance that we see everywhere in the body, or are there differences between different sites of cancer that acquire resistance and that might help teach us about the best ways to approach developing new therapies in this space. So far, this is the case that's been analyzed, and you'll see the different sites that were sampled from this autopsy case. And Dr. Lin hopefully will provide an update on the gene analysis of these different sites and give us really a picture of what happens in multiple areas that acquire resistance, in this case to selpercatinib. So samples were submitted for a look at DNA that's very comprehensive. We call that whole exome sequencing, and also a look at RNA, because we know that we may find different data depending on which nucleic acid we look at. All right, and in the last few minutes, I wanted to give an update on the second aim of the project, to highlight for you that there has been movement relative to when we first started this. If you remember, the question here was whether or not immune therapy is helpful for RET-dependent cancers. And we know that historically that unlike, say, smoking-related traditional lung cancers that may do very, very well with immune therapy, specifically immune checkpoint inhibitors that are approved, like pembrolizumab, atezolizumab, durvalumab. Many cancers that have a target in them, we've observed, can have poorer responses to immune therapy. And so you'll see our results here based on the registry data. These were patients that had not just immune therapy by itself, but immune therapy with chemo or chemoimmunotherapy. You see the breakdown on the left of what line they got the chemoimmunotherapy, and also on the right, their PD-L1 status. And you'll see on the right that there are some cancers that may have intermediate or even high levels of PD-L1, meaning 50% or greater. But despite that, you'll see here that the median duration of chemoimmunotherapy and the median progression-free survival was still modest in the order of 7 months. So 7.4 months on the left and 7.3 months on the right. So really the question here is chemotherapy by itself enough? And do we truly need to add immune therapy to chemotherapy? Now, before we circle back to that question, chemotherapy versus chemoimmunotherapy, I wanted to briefly show you that we had also looked just at immune therapy by itself. And you'll see the breakdown here of the different lines on the left and PD-L1 status on the right. And as with the historical data that we talked about, again, you'll see that the amount of time that patients were on an immune therapy was fairly modest at 4.7 months on the left and 5 months for progression-free survival on the right. So this brings us to one of the most recent updates, and that tries to answer the question that we had asked: is chemotherapy by itself enough, and or do we need to give chemotherapy with immune therapy? Does the addition of immune therapy to chemotherapy really result in substantial benefit? And the update for you guys is that we're very fortunate to have partnered with Lilly and Loxo to examine this trial, which I'm sure many of you are familiar with. It's called the Libretto 431 trial that looked at patients with RET fusion positive lung cancers that received either selpercatinib or chemotherapy plus minus immune therapy. And that's the curve in the control group that you see on the upper right. So while it was not the primary intent of the trial, we do have this data so that we can potentially see if the addition of immune therapy in patients that got it resulted in a dramatic benefit over just chemotherapy. And so we are currently at the stage where we have submitted all of the paperwork for the website that will allow Lilly and Loxo to give us all of the data on the patients that receive chemotherapy and immunotherapy. And we hope to do a comparison between the two groups to see if really you get much more time or benefit out of immune therapy or not, and marry that with the data from the registry. I will end very shortly, but the other thing I wanted to say was that Dr. Gainor, Dr. Jessica Lim have established nice data in another target, ALK, which they presented earlier this year at one of our lung cancer conferences, that showed that perhaps really the addition of immune therapy may not be helpful. So we'll see how this all shakes out. And with that, thank you very much for your time.
Thanks so much, Dr. Drilon. Now we are going to hear from Dr. Justin Gainor. So Dr. Gainor, whenever you are ready.
Okay, and let me just share my, okay, can you see my slides okay?
Yes.
Okay, great. I want to echo the sentiments, John, Alex, others of thank you so much to the Happy Lungs Project for really supporting this work and also bringing us together as a team. You know, when we sat down a couple years ago now to think through projects, these were all, you know, meant to be interconnected and focusing on different aspects of RET-positive lung cancer. And so, you know, you've heard a lot about targeted therapy. Alex introduced, you know, some aspects of immunotherapy. And so really this talk, as well as Dr. Reuben's talk, which will follow mine, is centered on, can we think about the microenvironment, that is where the kind of the surrounding cells of tumors and how that influences our therapies, and then can we use that therapeutically? You know, I think that when we think about immunotherapy, and Alex did a great job showing you that, you know, immune checkpoint inhibitors to date, you know, aren't providing the responses that we want to see in RET fusion positive lung cancer. But I bring this up to show you that, you know, just because, you know, that isn't the magic bullet, you know, it doesn't mean that all of immunotherapies are taken off the table. And really, we think about immunotherapy as a really broad class of therapies. And so just to walk through what some of those are, you know, on the left, you know, when we start thinking about things like cellular therapy, so Dr. Reuben is going to talk to us about efforts to develop kind of cellular-based therapies geared towards RET. And so I'm not going to spend much time on that today, but just to say, stay tuned, because that, you know, is going to be the focus of the next talk. But then when we start thinking about other things, you know, beyond immune checkpoint inhibitors, we have, you know, recent advances in anti-cancer vaccines. Cytokine-based therapies have been around for a while. They're certainly making progress in terms of the engineering for cytokines. These are basically like the signals that your immune system kind of uses to communicate with one another. And so can you actually like engineer in the lab cytokines? That I would say, in RET right now, I think wouldn't be my preferred strategy. But certainly, kind of thinking about advances in mRNA vaccines, and then antibody approaches. So I would say over the last 10 years, we've seen a lot of progress in engineering antibodies, not just so-called naked antibodies that just stick to one thing, but can we actually attach things to antibodies? Many of you may have heard of things like antibody drug conjugates, where we're actually attaching chemotherapy to an antibody, and the thought process being able to guide the chemotherapy towards the tumor to avoid some of that whole body toxicity. But also, strategies of taking antibodies and actually having one part of the antibody stick to the tumor cell, but another part stick to the immune cells, right? And so maybe that's a strategy of engaging the immune system. So we call those T-cell engagers. And I do think that over the next couple of years, we're going to be seeing more and more T-cell engagers and other things come out in the lung cancer space. We saw the first approval for a T-cell engager in small cell lung cancer earlier this year, and I'm hopeful that that's just the tip of the iceberg. This is just to show you that just with some more specificity, when we talk about these antibody drug conjugates, right now you can vary. So you can vary multiple things, right? You can vary what these stick to. So you can vary the antibody, and so different antibodies recognizing different things on the cell surface. So here showing HER2, HER3, TROP2, CEACAM5, c-MET. These are some of the more common antibody targets that are being developed in the lung cancer space. But then you can also start varying kind of what the toxic payload is. So what you're attaching to the antibody. And so, we know that these drugs have shown activity across, a large proportion of patients with lung cancer, but how can we actually get more specific? And are there certain genetic subsets of lung cancer that may be particularly enriched for responders to these types of therapies? And so with that as a background, as we start thinking about these different types of therapies, so then, it begs the question of what is when I say the tumor microenvironment, what actually exists within a tumor? It's not just 100% tumor cells, right? So, if we take this diagram here and we see it kind of the tumor cells in this kind of aqua color, we know that mixed within, we have various support structures. So blood vessels, we have things like fibroblasts, which are like those support type cells, depicted in this this green color here. Their role is to provide scaffolding, and we also have immune cells. There are many different types of immune cells, and so insights into what immune cells are present may actually help inform what types of immune therapies and other strategies we might use. And so, this is where I think, there's been, tremendous technological progress in the field over the last several years of how we've come to study tumors. And so I think this is really exciting. If we take, just to borrow an analogy here, right? 10 years ago, when we wanted to look at the RNA of a tumor, so imagine, say this berry basket here is the microenvironment, and we have, tumor cells, we have immune cells, we have those support cells. When we wanted to sequence it, essentially what we did is we ground it all up together like a smoothie, and you can't really differentiate any of the cell types, right? There are strategies to try to deconvolute this using fancy tools, but at the end of the day, it is it's a mixture of everything. of late, we've developed technologies where we can actually take individual cells within the tumor and actually sequence individual cells, right? And look at the expression profile of each individual cell. And so we can say, okay, that one's a blueberry, that, this is a strawberry, this is a raspberry, and we can actually look at the proportions of cells. And so that's one of the strategies that we've taken to try to understand what is the tumor immune interface of lung cancers. This is a plot here showing that, so far, we've actually done this in over 100 patients with lung cancer of various different gene types. Each kind of column here represents an individual biopsy. And so this is just a way to kind of aggregate a lot of the characteristics of this whole cohort. But what I wanted to focus on is, some of the observations that we've made, and just to show you the power of this technology and how we can then start using it to hone in and draw these comparisons, right? John gave you the intro of how do we start classifying things. And so I think this is a tool to allow us to start classifying things. So this is something called the UMAP, and I'm not going to get in too much into the technological details, but essentially, if you were to zone in here, every single dot on this plot is an individual tumor cell, is an individual cell, right? And so we can break this down, like in the brown, these are all the tumor cells. Over here in purple, these are the B cells, a form of immune cell, here are red blood cells, here are your neutrophils. So, this is across the entire cohort. And then, these are just high level definitions. We can actually get really, really granular here in that, this is just showing you different types of immune cells. You can get very, very, very specific. So again, each dot represents an individual cell. And so, we can now take this and say, okay, now let's pull out all of the cells within RET-positive tumor samples, right? And that's what this is looking at. So on the right here, so this is about 13,000 cells that have all kind of been individually sequenced from RET fusion positive lung cancers. A few things to point out. So in this kind of brown area over here, so these are the tumor cells. Okay, so you can see here that it's striking, right? Some of this has relates to the processing, but only a subset of the cells that we're seeing here are actually tumor cells. So what are the other things? So myeloid cells here in green. So these are types of immune cells. We have neutrophils, we have some T cells. So, right now we're taking a deep dive in importantly, because we can leverage that larger data set of over 100 patients, right, where we've also done this on tumor specimens, right, we can draw, comparisons between the two. So we have a large group of ALK positive, ROS1 positive, EGFR, and can we draw some comparisons? And that's really where I think we can start leveraging insights from other genotypes and can we actually apply them to RET as part of that. This is just taking that same plot and just applying some of the more specific labels here. So you can see even among those fibroblasts, the support structures, you can get, there are different types even within the same tumor. And so we can get really, really, really granular details. And then we can start asking questions. I recognize this may not project as well, but we can ask questions like, that have therapeutic implications, or at least framing the types of questions we're going to ask. So like in this kind of the same projection here, right? But now we're looking at, okay, we can actually take those plots and ask, what is the expression of one single gene, right? And so this is interesting. So if we just ask, okay, remember this area here, if you can see my mouse, is the tumor specimens, right? The tumor cells. And so we can ask, okay, do they express HER3, which there's an antibody drug conjugate in clinical development against HER3. So yes, some of the cells actually express HER3. What about MET? And so yes, some of them express MET. Doesn't look like we're seeing a lot of CEACAM5 expression. We actually are seeing a fair bit of TROP2 expression, right? That's that TAXS TD2. So I think that is a powerful feature of this technology. And then we just one of the challenges is it's requires real-time processing and a fresh biopsy, so it's not something that you can easily go back to up until recently old biopsies and do this. But what we did is we followed this up and actually doing standard stains and start looking at those same antibody drug targets that I mentioned, and we actually see each row here represents a different patient, and this is just how we score, how the pathologist scores it, but we actually did see a fair number of RET-positive tumor cells expressing MET, and there are antibody drug conjugates in clinic, TROP2, same thing, and so I think we want to now take this forward and apply. There are different ways where we can actually start applying these same technologies, these spatial technologies, but preserving the tumor architecture. So in a biopsy and seeing what the relationships are in space. And some of the ways we can do this is what's known as multiplexing. So you basically taking multiple antibodies that are have different fluorophores, so they respond differently to different wavelengths of light, right? And so you can put multiple antibodies on a slide and you flash different wavelengths of light, and you can actually start generating pictures like this, where you can actually look at what's the relationship between multiple targets within a single tumor. And this is really helping us, I'll kind of speed through this a little bit, but really helping us determine, like, okay, there are some tumors that maybe are particularly immunotherapy responsive in lung cancer, defined by these hubs. It gives us a much better definition of what's happening within the neighborhoods around the tumors, rather than our simple kind of assessments of one marker at a time. And so this is some work where John Chen, working in Near Cohen's lab, had shown that these tumor immunity hubs really are good markers for at least immune checkpoint inhibitors, and so we looked at these in RET fusion positive lung cancer, and so this kind of aligns with what Alex just showed you, is that we weren't really seeing a lot of these hubs in RET, and so this really, in my mind, underscores, one, we need to think about strategies. How can we induce that and perhaps make checkpoint inhibitors work better? Or separately, I showed you the data on antibody drug conjugates, and perhaps instead of either attaching chemotherapy or these T cell engagers, and really try to bring the immune system to these tumors. I'm close to time, so in the last minute or so, I did want to just mention one thing about the fibroblasts. I started by saying, these are some of the support structures of the microenvironment, and so historically, whenever you're building models, you try to get rid of the fibroblasts when you're trying to create a patient model of a tumor. But Aaron Hata in our group started studying and actually finding that the fibroblasts have their own characteristics, and that the fibroblasts actually, if you actually culture the fibroblasts and mix them back with the tumor cell, as kind of mimicking what is happening within the tumor in the body, that sometimes the fibroblasts actually send growth signals that make our targeted therapies not work as well, right? And or sometimes influence the immune microenvironment. And so that's what we've also been doing, is in addition to efforts to try to create cell line models from biopsy specimens of RET-positive lung cancers, in parallel trying to create fibroblast models to try to capture some of those support structures. And this is just to show you on the left that it actually does make a difference. So in gray, John introduced this concept of like these curves when you apply a drug like selpercatinib to the cells, and essentially in gray is just if you're just giving the cell line selpercatinib alone. But if you co-culture it with the fibroblast, you actually find that it becomes less sensitive to selpercatinib, right? And what we see on the right here is actually, if you look at multiple different fibroblasts, they actually have very different properties. Some actually really cause marked decreases in sensitivity, while others not so much. And so trying to understand what are the key factors that the fibroblasts are secreting that are causing this behavior, because that too can inform some of our strategies. So with that, I wanted to thank everyone for their attention. This is actually the work of so many people. I want to call out specifically Jessica Lynn from MGH, who really has driven a lot of this work. Mari Mino-Knudsen, also one of our pathologists, and thank everyone on the call today and the Happy Lungs Project, really, who have really enabled all of this work. So with that, I will pass things back.
Thanks so much, Dr. Gainor. And last, we are going to hear from Dr. Alexander Reuben. So, Dr. Reuben, if you're ready.
Thank you, Rennie. Hopefully you guys can see my slides.
Yes.
So I appreciate support from the Happy Lungs Project. Thank you to John and to Alex for introducing the space, and to Justin for introducing a little bit more about immunotherapy. Our lab is specifically focused on cell therapies, so a subset of immunotherapies and how those could be applied to RET cancer. So we know quite well that the immune system does protect us from cancer on a daily basis. Probably the simplest experiment you can do to test this is that if you take mice with and without an immune system and you expose them to chemical carcinogens, over time you'll see that the mice that have no immune system will develop cancer to higher levels and much more quickly. But you do see that the mice, even with an immune system, do eventually develop cancer here. Now, in immunology and tumor immunology, we have this concept called immune editing, and there are 3 phases to this. The first is called the elimination phase, and this is a phase during which the immune system is able to effectively see and destroy tumor cells. That's followed by this equilibrium phase, where there's kind of this back and forth between the immune system and cancer, and eventually the cancer takes over, and the immune system is no longer able to see or destroy these tumor cells. And it's at this stage that many of the mediators of this process are the focus of different therapies. So if you actually take a tumor and you slice it and you look at what immune cells are present, this is what it can look like. Each one of these colors represents a different type of immune cell. So you can see there's a lot of immune cells in these tumors. If you focus on only one of these types of cells in red here, these are the cells that are called T cells. And these are the cells that we focus on primarily. And the goal of these T cells is really to see and destroy any cells that are infected or that are malignant. But as you can see, obviously that's not happening in this case, despite the presence of a large number of T cells. So T cells can see cells based on a process that's called antigen presentation. So all of your cells at all times are doing what's shown on the left here, where basically they are displaying at the cell surface proteins that are reflective of the state that they're in. So if one of your cells is healthy, it's displaying healthy proteins or antigens at the cell surface. If your cell is infected by a virus, well, there's viral proteins at the cell surface. If it's a malignant cell, well, there can be cancer antigens at the cell surface. And this is what helps the immune system, and specifically T cells, scan each one of those cells and determine which ones should stay and which ones should be destroyed. So another way to look at, excuse me, at this process of antigen presentation is that this is kind of like the ID badges of these cells, right? So each one of these cells has one of these ID badges that allows the immune system to determine whether they should be there or whether they should not. And so for these different cells, when the immune system, specifically T cells, recognize these antigens or see these ID badges, then they'll be able to take appropriate action. So a T cell will ignore a cell that's healthy while destroying a cell that's malignant or that's infected by a virus. And the way this happens is through what's called the T cell receptor, which is drawn here. And there needs to be recognition of the antigen as well as what's called an HLA molecule that is basically presenting that protein to the immune system. And as a result of that interaction, then you have what happens on the right here is that the T cell can become what's called activated, and that leads to release of different proteins that can lead to cell death and the killing of tumors or infected cells. Now, as I mentioned, HLA molecules are extremely important when we're talking about this form of cell therapy. If you have 6 different patients that have the same mutation, well, that could potentially be targeted, but only if they have the same HLA molecule. And that's because of the shape and the structure of those HLA molecules and their ability to present those antigens. So as you can see in this example, despite these 6 patients having that same potential antigen or mutation, well, only the ones that have the same HLA will be able to effectively display or that protein to the immune system. And so here's a video showing what can happen when a T cell sees its antigen. The T cells are in pink, the tumor cells are in green. And as the T cells kill the tumor cells, they start to shrivel up, they detach from the plastic, and they become red because basically their membrane is becoming permeable and they're able to incorporate this dye that turns the cells red. So T cells are extremely effective, but the question is, how do we make them as effective as they can be? So one form of cell therapy is the adoptive cell therapy using tumor-infiltrating lymphocytes. And the way this works is that typically a tumor is resected, surgically resected, it's removed from the patient, it's chopped up into small pieces, and then the T cells or the immune cells that are present within that tumor are grown in the lab and then reinfused into the patient into the tens or hundreds of billions of cells in order to eradicate what's remaining of the cancer as well as protect from any recurrence. And this can actually work pretty well. These are 2 studies from the last few years in lung cancer. Each one of these lines represents a patient, and you can see that there's some patients that have complete responses to these treatments where their tumors just completely disappear and melt away. But it's much more of a mixed bag for other patients. Some patients don't benefit at all, and some initially benefit and then develop new tumors. And so the question is, how do we make this as effective as possible? Now, one of the issues with treatment with adoptive cell therapy, and TILs in particular, is that the cells that are inside the tumor, they're not always effective at seeing and killing that tumor, as is shown in this plot here. Basically, what this is telling you is that there's only about 15% of those cells that are able to see the tumor, and the vast majority are actually not able to do so and may actually be detrimental. In the middle here, this is what's called an ELISpot, and each one of these spots represents a T cell that's secreting a cytokine or becoming activated upon exposure to a different type of antigen. And what you can see is that these tumor-infiltrating lymphocytes, a large proportion of them actually recognize what are called viral antigens. So these are antigens that are arising from influenza, CMV, and other types of infections. And so these are cells that you would not expect to be in any way helpful in eradicating a tumor. And what happens when you take those cells and you put them in contact with tumor cells is nothing. The T cells are in green, tumor cells are in red. And despite the best efforts of the T cell to see and kill the tumor, as you can see, there's really nothing happening here. And actually at the bottom here, the tumor cell is actively dividing as the T cell tries to kill it. So here's other illustration of this process. On the left is a scenario in which you have the right T cells present. On the right, the wrong T cells are present. And so if you have the right T cells present, obviously you're in good shape because your T cells are able to see and destroy the tumor as is shown here. What if you're in the situation on the right here where the right T cells are not present? How can you actually get T cells that will help you eradicate that tumor? Well, that's when we look to what's called cell engineering. And you may have heard about CAR T cells. I'm happy to discuss those briefly, but our focus is on what's called TCR engineered T cells. And these are cells in which we are engineering or inserting a gene that allows these T cells to actually see new proteins that they were not previously able to see. And the advantage of a T cell receptor as opposed to a CAR T cell is that whereas a CAR T cell is only able to see what's displayed at the cell surface, a TCR can theoretically see anything regardless where it's found within the cell. And so that kind of opens up our options here. This is just a table showing some of the different cell therapies and some distinctions. And as I mentioned, I'm going to be focusing on TCRs here. And so the thought is that if you have all these cells that are circulating, that are recognizing different types of infections and viruses, when they're encountering a tumor cell, they really just won't care. They won't be able to see that tumor cell. They won't be able to kill the tumor. And so what you can do is you can carve out these receptors and replace them with receptors or T cell receptors that are now able to see and kill tumor cells. And so what you have now is a supercharged, army of T cells that are able to destroy your cancer. And so what we've done over the last several years is to develop a platform that allows us to do this. I won't go through the details, but essentially it allows us to do the discovery of these T cell receptors, as well as their validation through several methods that are outlined here. Now, when it comes to RET fusions, as you heard, these tumors are not very responsive to immunotherapy, as you can see here. Part of the reason, as was described by Alex Drilon, is that there's not a lot of PD-L1 in many cases on these tumors, and PD-L1 is actually the target for many of these checkpoint inhibitors that are using antibodies. The other reason, or one of the other reasons, is probably the low tumor mutational burden, and that's important because these mutations are actually what makes the tumor visible to the immune system. The more of these there are, the more foreign the tumor looks to the immune system, and the more visible it is. And so despite the low presence of mutations in RET fusions, there may actually be antigens that are actually relevant to cell therapies and that we can focus on. So immunotherapy, as I said, really focuses on the concept of comparing self versus non-self and the ability of immune cells to distinguish between healthy cells and infected cells. I won't go through all of these different types of antigens, but we have efforts targeting or attempting to target many of these in the context of RET. Now, with RET fusions, as you saw, there are many different fusion partners that exist, including KIF5B and CCDC6. Initially, what we did was to identify potential peptides arising from the fusions between those two proteins to try to test whether those fusions are what's called immunogenic, or whether that makes them visible to the immune system and can lead to tumor killing. And in addition to that, we had an approach that included a couple other strategies. So as I mentioned, we targeted the fusions here arising from the intersection between both of those genes. We also targeted what are called epitopes arising from RET that are associated with the function of RET, so what's called the tyrosine kinase domain. And then we also looked for other proteins that are high in RET tumors that we could be targeted through cell therapies. So here's an example of some of our data. This is what's called flow cytometry. Each one of these dots represents a tumor cell. And what you can see is that as you look to the right in that red box, these are the tumor cells that are actually being killed by the T cells. And what we saw was that when we looked at blood from many different blood donors, we were actually able to identify T cells that are able to effectively kill tumor cells. And the validation of these T cell receptors is currently ongoing. Now, we've also been looking, as I said, for proteins that are found within these tumors and that could be targeted by the immune system. Here's an example of one such analysis where the tumors with RET fusions here are shown on the right. And you can see that there are certain protein or certain RNA transcripts in this case that are found at much higher levels in these RET tumors than they are in any other tumors. And importantly, not only are these tumors expressing higher levels of these proteins, but these proteins are actually found at even higher levels upon resistance, as is shown here. And this is data from John Heymach's lab. And so we now have been focusing on developing receptors that target those proteins. This is a killing assay where over time you can see that as the line goes up, that means that there's more and more tumor cells that are dying. And so this is exactly what we would like to see as we validate these therapies. We also perform what are called alanine scans, where we're mutating the different parts of the antigen to determine exactly which parts of that antigen are required for the immune response. And this actually can be indicative of proteins that are good versus bad targets and help us identify potential targets that could lead to toxicity and cross-reactivity. And so as a result of this work, we've now validated 5 different T cell receptors that are targeting these proteins, and we're working on trying to bring these TCRs further into the clinic currently. So that's all I have for you. I want to highlight the people that actually did the work here, some of our collaborators. I want to once again thank Happy Lungs for the support, and also RET-positive, who's been very helpful in helping us bring cell therapies to RET patients.
Thanks so much, Dr. Reuben. Now I'm going to turn it over to Ilana Stromberg. I think you are muted, Ilana. 1 second. Let me.
There you
Yeah.
go. So
Yeah.
sorry. Hi, I'm Ilana Stromberg. I'm a board member and co-founder of the Happy Lungs Project. I'm also a RET lung cancer survivor, like many of you here today. Thank you again, Dr. Heymach, Dr. Drilon, Dr. Gainor, and Dr. Reuben for your fascinating presentations. We appreciate you taking the time to update us on the research and answer questions in the chat. I'm feeling really encouraged about the advances and hopeful that we're going to continue to make great progress. We'll address all the other questions that weren't answered today, including the ones that were sent to us prior to the meeting today following the webinar, and we'll get those answers back to everyone as soon as possible. As we close today's webinar, I just want to thank our friends at Lilly for making this possible. We're really grateful for your support of the RET community. I also want to thank everyone who attended today. We will post a recording of our website on our website, and you'll receive a follow-up email with the link sometime next week. Thank you all for being so supportive of the work of the Happy Lungs Project and our mission to support researchers and clinicians like those we've heard from today in their work to find better treatment options for all of us. The research only happens because you donate. During this holiday season, please consider supporting the Happy Lungs Project through a donation of any size. Here's the link and a QR code that leads to our donate page. Thank you again for joining. Have a great day and a wonderful Thanksgiving holiday. Be safe and healthy. I am thankful for all of you here today. And now we're right at the end. If anybody has any other questions or anything they want to add, we've got about 5 minutes. Otherwise, happy Thanksgiving and thank you.